else, thank you so much for joining us tonight. I can already tell this is going to be a fun, dynamic conversation with all of you. My name is Guillaume Urtecho. I am a scientist in Harris's laboratory who's going to be talking later. I use he, him pronouns. And I'm really excited, I've been doing DNA synthesis, I guess, for the last, it was my PhD work. And so I've been at it for like 6 years and love it. I think it is the technology of the future. And I'm really excited to hear your thoughts on it and what we can use if we can write DNA and hack the blueprint of life to create whatever we want. And today, the outline of this seminar is going to be, first, we'll start off with a little background on DNA synthesis and in the context of the blueprint of life. So what is this blueprint and how do we write DNA and how can we use this to modify the blueprint? We'll have some time for questions and answers, specifically afterwards, but feel free to hop in whenever you'd like. If there's a slide you have a question on or just an idea like, hey, wait, why don't we do this? We'd love to hear it. So please feel free to leave a comment in the chat or just unmute and let rip whenever inspiration strikes. Then after we've gotten a little technical understanding, we'll hop right into really the 20 years into the future. What are applications of being able to synthesize DNA? And what can we build? And also, what should we build? Are there things that we maybe shouldn't? And this section, we're going to integrate a lot of polling to get a sense for what you guys think are the right choices we should make and the right questions we should be asking. And then we can hopefully come to terms with all of these concerns with a final Q&A. But first off, I was talking about this blueprint for life, and what I was really meaning was DNA. This is an incredible molecule which is found in all organisms, and it encodes the information necessary to create these various forms of life. So what's really amazing about this molecule is how simple it really is. A DNA molecule is just a long string of these 4 different base subunits known as nucleotides. And when these nucleotides C, G, A, T are strung together in specific ways, cells can interpret these sequences to enact biological functions. And while to us, a DNA sequence looks like just a random string of these letters all mixed up, to cells that are able to understand this language, they can use these sequences as the instructions necessary to create life. So how many letters are needed to create life? How big is that instruction manual? Well, when we talk about the complete set of DNA necessary to construct an organism, we're talking about that organism's genome, the complete set of DNA in a cell. So genomes are, of course, made up of many genes, which each encode a different protein or biological function. And these genes are on average 1 kilobase or 1,000 DNA letters in size. Now, just a bacterial genome, which contains thousands of these genes, can reach 10 million base pairs in size, so 10 million letters to make a bacteria. And mammals, including humans, they contain tens of thousands of different genes, and so require billions of nucleotides to create these genomes and encode all these various functions. Tons of information. In fact, in just a single human cell, if you were to take out the DNA and stretch it out end to end, this would reach 6 feet in length. And if you were to take all of the DNA in the human body and stretch it out, this would be a ridiculous 67 billion miles of DNA, which is enough to stretch from the Earth and to the moon 150,000 times. So tons of information is required to make biological organisms. So now that we know what the blueprint is, what can we do with it? Well, it turns out we can do a lot of the same things that we do with a blueprint in real life. We can read these blueprints to learn how things are made. We can edit these blueprints to make changes and move things around. And we can also write these blueprints from scratch using DNA synthesis, which is the focus of this seminar and why we're all here, hopefully. So first, how do we read the blueprint? Well, the technology to read DNA is known as DNA sequencing. And this technology first became big in 1990 when they announced the Human Genome Project, which, as the name implies, sought to sequence the entire human genome. And this project was a huge success. It was, as far as government projects go, it was somehow on time and under budget, taking only 13 years and $2 billion to complete, which, still is a lot. But since then, our ability to read DNA has frankly improved to levels that are approaching science fiction. And now we can sequence a human genome for just about $1,000. That's so going from 1 billion to 1,000 in 20 years. That's a 1,000,000-fold improvement.
Oh no. Rename.
Oh, hi, Theo.
There we go. Yeah. Sorry. Thank you.
So yeah, when have you ever heard of anything becoming 1,000,000-fold cheaper? It's absolutely astounding how powerful these technologies have become. And fortunately, we've made great use of this powerful technology. Over 1 million human genomes have been sequenced, allowing us to learn about how biological functions are encoded in our DNA and how genetics control human disease. There are even commercial products that you can purchase to take minor glimpses into our genomes, as well as our pets, and as well as our pets. Although, if you look at this cat right here, I don't think they appreciate this as much as you might. So it's great that we can read it and learn how information is encoded in biology, but the next step is being able to make changes to that, editing that blueprint. And we've known that this blueprint is capable of changing. In fact, nature edits this blueprint constantly through processes of evolution and natural selection. This was first documented by Charles Darwin in the 1800s, where during his tour of the Galapagos, he realized that the beaks of the finches he was studying seemed almost designed for eating different types of food that were found around the islands. And he reasoned that a process of natural selection had changed these birds, changed the blueprint for how they are made. And so even though that was from the 1800s, we actually only just recently learned what that change is to these blueprints. And it turns out that there is one gene, the ALX1 gene, which largely determines beak shape for these different birds. And so birds that have different beak shapes also have different instructions at this gene. And this is an example of a monogenic trait, which is a trait that is only controlled by one gene. Similar to the ability to taste phenylthiocarbamide, which if you have the wrong gene, or I say it's the wrong gene, then cilantro will taste like soap, which is very unfortunate. And if you don't have the gene, then you get that nice, refreshing, like little, I don't know what to call it, it's great. And of course, there are also polygenic traits. These are traits that multiple genes can control and affect. Height is the most common example of that. There are many genes involved in it. So how is evolution changing the blueprint? Well, it does this through small random changes over time. And the most common changes that we think about are mutations, first off. So every time you copy your DNA and pass it on to the next generation, inevitably that creates errors in the copying. These can be letters inserted, or letters deleted, or letters changed to a different one. And those are pretty small. However, you also have these more drastic changes or recombinations. Remember, we have chromosomes from our mother and father, so we have pairs of chromosomes. And inevitably, these chromosomes are capable of shuffling DNA between them, creating new hybrids of these chromosomes, which creates an incredible opportunity for genetic diversity. And with all these changes happening, gradually you see the blueprint for organisms change. So although from one generation to the next, you'll only see small changes in the blueprint, After many generations, these changes start to accumulate and you get vastly different types of organisms. And yes, this is still happening in humans today. At least in humans, between generations, we see about 1 to 100 mutations, and also 20 to 50 different chromosomal recombinations. So we are changing, and who knows what humans will be like in thousands of years. So long before we knew what natural selection was, we've actually been able to exploit it in order to edit the blueprint of various forms of life in order to, or for our favor. And this is a process called selective breeding. One of the major examples of this is the domestication of corn, which took place in southern Mexico over 9,000 years. Over here is the ancestor of corn, teosinte, which frankly does not look tasty at all. If you bring this to your 4th of July barbecue, you're going to get turned away at the door. However, because ancestral farmers constantly bred corn that had larger and larger kernels, larger cobs, this has resulted in the tasty side dish that we know today. And this is still happening even very recently. Brussels sprouts actually in the 80s tasted awful. And some team from the Netherlands thought that won't do. And so they started this huge breeding program to create tastier Brussels sprouts. And these are now the dominant variety on the market. And, you know, honestly, pretty good. If you haven't had one, I suggest you give it a try. And of course, the most famous example of selective breeding is the domestication of wolves into our canine companions. And we've done amazing things to wolves, not just in how they look, creating these very cute forms that we see here, but also how they behave. So you can change so much about biology through the selective breeding. Now, the way this works is that first you have 2 parents that have desirable traits or 2 good blueprints, and then you breed these. The next generation is going to be a mix of characteristics from the parents, but different combinations essentially. And from those different combinations, you can identify those that have the most desirable traits and then go on breeding them to create the next generation. And then this process can be repeated and repeated until you have reached the desired outcome, which can actually be a blueprint that is very different than what you started out with. And lastly, we also now have very modern ways to precisely edit the genome with very sophisticated molecular tools that are at our disposal. And many of you have probably heard about this CRISPR or Cas9 and its potential for gene editing. If you watch NCIS, they talk about, you know, CRISPR freaks sometimes. It's a little ridiculous, but this is a very powerful molecule. But you might be surprised if you didn't know that this is actually an enzyme that is from bacteria and their immune system. Bacteria use this enzyme to target DNA from viruses and cut those viral DNA into pieces before the virus can infect the cell. And the way the system works is that you have your Cas9 enzyme, and then this enzyme is provided with what's called a guide RNA, which is a RNA sequence that matches a section of the target DNA, which in the case of bacteria it'll match a viral DNA. And then when you give Cas9 the sequence of the guide RNA, it finds that sequence in the genome and then can act on it. Primarily, it cuts it. And Jennifer Doudna and Emmanuel Charpentier realized that this was a very powerful tool, ability to target DNA sequences. And they have found many different ways to repurpose it, which is why it got a Nobel Prize in just under 10 years since its discovery, which is absolutely absurd. Most things take a long time to realize their potential. So what is that potential? Well, if you can target DNA, you can pretty much target various different edits to those sections. And with Cas9, we're able to edit single bases, make small changes, editing just single bases at a time. We can also make large deletions in DNA sequences that we don't want, or even insert brand new sequences, adding new functions, new DNA to do like things that you'd normally never see in a genome. So very precise changes and a lot of different options. That's why everyone is so excited about Cas9. So while we can do pretty incredible things using DNA editing, the shortcoming of all these methods is that you're only able to act on existing sequences. You can only edit a DNA sequence that is already exists to some capacity. On the other hand, if we're instead able to write DNA, you can write any DNA sequence. You're not restricted by any means and can create completely new DNA molecules from essentially nothing. So how that looks is that with DNA synthesis, you're starting with a complete blank blueprint that you're able to write on using these different nucleotides. And by adding different nucleotides and creating new genes and new genomes, you can create pretty much whatever you want. And it doesn't even need to be things that exist in biology. If you can build it, if you can think it, you can build it to an extent. However, with this much control, it also is much more complicated, and you need to start thinking about what should you design, how much would it cost to build, and how accurately can you actually build it. So now let's talk about how writing works. How do we write DNA and the blueprint? Well, even though we're talking about its potential for the future, it's actually been around for a long time. Here is a real receipt from a DNA synthesis order in the 1980s. Back then, and DNA synthesis was just starting to get into fashion. But it still required highly skilled organic chemists to synthesize sequences. And this process would take many weeks and cost thousands of dollars for very, very small sequences. Here the bill is $6,700 for 10 bases of DNA, which, remember, a gene is like 1,000 bases. So it would take 100 times as much money, what is that, $670,000 to create a gene back then. Which is definitely a no-go. However, there have been huge advancements since, mostly in the form of automation. And now we have, instead of your cranky old organic chemist, you have these very sophisticated DNA printers. This is a video of a printer from Agilent, which is, we're talking about DNA printing. Agilent is actually owned by Hewlett-Packard. So, your favorite printer company. Now they have a DNA printer. And here we're seeing each of those different spots is a DNA sequence, and the printer is going around adding new nucleotides, new letters to that chain, extending that DNA molecule. And look, it's able to synthesize each of those spots is a different DNA sequence. So you're synthesizing hundreds of thousands of sequences at a time at really fast pace. And when you have these technologies, it brings the cost way down. From $6,400 for just 10 bases, now for 500 bases, you can get 15 million nucleotides worth of DNA. So huge jumps have been made. Now, that video of what we're seeing here was an example of phosphoramidite chemistry, which is the current gold standard. And honestly, it's been the gold standard for about 40 years. So the way this works is that you start out with your synthesis platform and add the first base through the process of coupling to that synthesis platform. And you can do many molecules at once, remember. These bases contain these caps, which stop addition of extra bases. Otherwise, you'd have no control of what base gets added next. So once you've added these bases, when you're ready to add the next one, you simply remove the block or the cap, process known as deblocking, and then this frees the molecule to be able to add additional nucleotides. So you add a nucleotide, remove the cap when you're ready, add the next one, repeat and repeat until you've created your desired nucleotide sequence. So the shortcoming of this is that every time you do this, it's only about 99.5% accurate, which sounds super accurate. But when we're talking about doing this 200 times, the probability of making a mistake becomes actually pretty likely. So here we're looking at the probability of not making a mistake depending on your length of DNA. And you can see that for 100 base pair sequence, only about 30% of your molecules would be created correctly. And for a 200 base pair sequence, you'd only make it right 10% of the time. And now this sounds, so the longer the sequence is, the less likely you are to actually create it. And while this may sound concerning, we're actually able to make it work, and I'll show you how in a few slides. But first,
there's a question about what causes the error.
Yeah, so why does this error occur? My understanding is that it is mostly involving this deblocking step. Sometimes the molecules actually don't have the cap. So you add a molecule that doesn't have a protective thing, and then something else can add on that you don't want it to. But the most common thing is that you actually fail to remove this. So you repeat the cycle, but since it's still protected, you can't add anything else. And you essentially skip one of the nucleotides, one of the letters. But so if that deblocking stage is an issue, maybe there's ways where we can get around that. And that is part of the future of writing DNA. And this process, which has gone from phosphoramidite synthesis, now people are excited about enzymatic DNA synthesis, or using enzymes to facilitate the DNA synthesis process. And all these technologies revolve around this enzyme called TDT, which is actually used by mammalian immune systems to create new antibody sequences in order to identify different viruses that are constantly evolving. So if it's good enough to create new sequences in our immune system, it should be good enough to make DNA sequences in our labs. And this works the same way. So you have a DNA sequence, and then your TDT enzyme containing the next base that you want to add. This TDT adds the base while simultaneously protecting it. And then when you're ready to add the next one, you just do the deblocking, deprotect to remove the TDT, and you have your sequence plus the base you added. And then as before, the cycle is repeated. And supposedly with this method, your accuracy goes from like 99.5 to 99.95, which actually ends up being a pretty huge jump. And there's a lot of excitement for this, the ability to create longer DNA sequences with fewer sequence errors and more power to create things in biology. And that's why there's a bit of a race going on, actually. Many different companies are developing slightly different technologies to get this to work. But for now, it's still up in the air as far as which one is going to be the next dominant competitor going forward. So, we talk about DNA synthesis and how it does have some shortcomings, but what can we create, despite all this? Well, first, I'd just like to highlight the incredible challenge ahead. So remember, we look at genomes. Bacteria is millions of bases, and a mammalian genome is billions of bases. And DNA synthesis, with oligonucleotides, gives us 300 base pair molecules. So, there is some disconnect. How do we go from 300 base pair molecules to like millions or billions? It seems like an impossible jump. However, we are actually able to do this, and it's through a process called assembly, which works a lot like how you build something out of Legos. You don't just get a chunk of whatever you're trying to build, but you first get these individual parts, which you can put together until you've created the fully sized object. And DNA synthesis and assembly works the same way. You have these small pieces or oligonucleotides that you stitch together into genes, and then you combine those together in order to make genomes. So it's a stepwise assembly process that you can kind of see here, where all these parts are arranged together in specific orders in order to create much larger sequences that you can tether together.
Before you go on, Gwen has asked a question in the chat. How do you specify the base that gets added with enzymatic synthesis?
Hi, Gwen. Great question. So how do you specify the base? Well, they have different TDT enzymes that are each loaded with different nucleotides. So they'll have the A, the C, the G, or the T combined to it. And so you just add when you're ready to add the next base, you just use the appropriate enzyme.
And is that mostly done through like microfluidics now? Like, do they kind of wash the enzymes in as they go, or what's the, yeah?
I'm not so sure on the chemistry if it's still solid phase. It's definitely, would I consider it microfluidics? Yes, but I don't know. Does microfluidics imply emulsions? And I don't think any emulsions are involved.
Right. And then you got some follow-up questions from Leandra. Leandra, are you in a spot where you can unmute and ask directly, or would you like me to read them out?
Oh, I can unmute. So I guess the components, the ACTGs, where do those come from? Like, do they get them from existing DNA? Have they like taken them out somehow and like stored them somehow? Or are they like, is it possible to create those from scratch?
Yeah, that's a great question. I'm pretty sure these are like mass produced by bioreactors. So they have bacteria or something that are producing large quantities of these that can be purified, or at least large quantities of a substrate that can then be chemically altered to produce these different molecules. So maybe Harris has has a better idea for that.
Yeah. Oftentimes there are chemical synthesis, methods to generate the phosphoramidite chemistry. Really interesting story is actually during the Beijing Olympics. They had to shut down some of the factories that use some of the chemical precursors for this, and there was a global shortage of the phosphoramidite chemistry. And IDT, which is one of the premier kind of DNA synthesis companies, almost went out of business because they essentially couldn't run their entire kind of business because it has no kind of precursor for it. And they eventually figured out another way from a petrol-based source, but it's actually it's an industrial scale. They're kind of byproducts that could be used as inputs to generate that.
Cool, thank you.
Right. So when we have these small molecules that we synthesize, we're actually able to assemble them together in order to create much larger genes and even whole genomes. Well, are we actually able to do this? I'd say the answer so far is like yes, and we're getting better every year. So the first like prominent DNA or genome assembly was poliovirus. This was in 2002. Where they assembled 3 fragments in order to create the 7.5 kilobase or 7,500 base letter genome. 8 years later, we've created, or this group from, I believe that's Craig Venter's group, created the first bacterial genome. This is 1 million bases large and was done by assembling thousands of different cassettes into much progressively larger and larger pieces. As we mentioned, it's a stepwise process. And actually fairly recently, it's quite the jump, but there is an organization out there that is trying to synthesize the human genome. So there was the Human Genome Project originally, and now we're on to the Genome Project Write, which is to write or synthesize the human genome. 6.4 gigabases. And, you know, expect this to happen, I would guess, before 2030, honestly. But it definitely does have technical challenges ahead. So to summarize, DNA synthesis, it doesn't necessarily allow us to synthesize an entire genome, but it allows us to create these building blocks for life that we can then assemble in many ways in order to create different things that, you know, already exist in biology, but also maybe have never existed before at all, to an extent. You know, with this, you have immense potential to control biology, and, you know, honestly, a lot of power that we should really consider at going forward with these, especially as technologies get better and better. So that's the end of part one. Going forward, or we'll have a short Q&A if there's any more questions. And then Harris is gonna talk about, so if we can synthesize life, what are we doing, and what is in the future of DNA synthesis? And also, you know, what should we be doing?
I'm going to pause the recording so folks can feel free to turn on their videos and say hello.
All right, do you see my slides? Okay, great. So I'm going to take over for Guillaume. That was a great primer there. I'm Harris Wong. I'm an associate professor in the systems biology department at Columbia University at the Medical Center. And so today in this part two, I wanted to kind of go through some of the applications of synthesis that are kind of either emerging or have been, you know, brewing in the last 5 or so years. You know, love to talk with you about both the possibilities, the you know, the applications of those, and you know, other types of interests and concerns that people have. And you know, those are kind of be broken down along the lines of, you know, storage of DNA as a potential replacement for hard drives, in the context of pandemic preparation and studying those, as well as generating new organisms with different properties, and rewiring kind of metabolism for various kind of applications on Earth as well as in space. So, and definitely do stop me at any time for clarification or comments and thoughts. The first of which is along the lines of information storage. And so if you actually look at kind of, you know, in the digital era, right, we have so many different mediums, you know, especially recently in terms of how we store more data. And if you kind of work backwards all the way from compact disks to floppy drives to tapes, and then even kind of books, right? There, you know, we've had a long history of you know, storing data and human history essentially in some physical medium. And as we're kind of building up more and more data in this kind of digital era, this is just a projection of how much new data is generated. You know, a zettabyte here corresponds to 1 billion terabytes. So that's a terabyte is, you know, the size of a hard drive, you know, in your computer, and now you have 1 billion of this. This is per year, right? And so the amount of information that we're generating is just tremendous, and our ability to really back them up is I would say, much worse than that, or you know, it's much more limited comparatively. And so I think, you know, DNA storage is definitely a really interesting opportunity that leverages this, you know, technology for synthesis. And I would say that has a potentially a much more important context in that we want to be able to preserve information moving forward, right? You know, we have books that we have, you know, we can go back to from, you know, maybe 1,000 years ago, right? But if we don't have those books anymore, you don't actually know anything about what happened then. And actually, if you look at like the Dark Ages And, there was not only kind of there's all these issues in history, but it's actually just a lack of information. And I think, just thinking about that, being able to generate and back up kind of the data on a regular basis is really important, not just for keeping historical records, but keeping them accurately so you can archive them over long periods of time. And the argument for why DNA is such a great information storage medium is actually that it's three-dimensional, right? Compared to all the other information storage mediums that we have in terms of hard drives and flash drives. They're right now mostly all two-dimensional architectures, which means that you're limited by the surface areas, right? But in DNA, it's in 3 dimensions. You can be folded in 3D, and so that extra dimension gives you so much more. The other thing is that it actually requires a very limited amount of energy to power DNA. A single cell has the capacity to copy its genome, every 20 minutes. Right, and so the power usage of this is very high. But probably more importantly, it's just that you can retain this information for potentially decades or thousands of years. And certainly we've been able to sequence DNA from organisms that have died from a long time ago, and so that information is actually very well retained. Which is not something you could say about flash drives or hard drives. And when's the last time you would feel confident about pulling out a 3.5 inch hard drive and hoping it will work, right? And so and it's also that it's not subject to this idea of technological obsolescence, right? So DNA is always going to be around. We will always have better technologies to read it. And so therefore that should be the universal storage medium. We should not be like going back and forth and continually recopying information that was stored from one medium to another, right? I have files from when I wrote my essays or my doing my work from when I was a graduate student or an undergraduate. I basically have very limited ability to extract that information, not because I don't know what the physical material is, because I don't have like the zip drive to be able to actually download that information now, right? And this is going to be true in the future. And so how this would work in the context of DNA is that you could digitize or binarize any digital information as it normally is done in a digital format, but then be able to encode that into nucleotide bases. And of course, here you have 4 letters instead of 2. And so you actually have a lot of room to build in redundancies and to have error checking. And then you can be able to synthesize this in bulk to be able to then store this physical material. And these are some kind of recent examples of people actually trying to do this. And so this was a book that was described from George Church, who was actually my PhD advisor back at Harvard. And he had written about this idea of storing information, digital information in the context of in DNA form, and then actually putting that into pages within the book. And so this is within this little piece here. He has hundreds of thousands of copies of this book. And when you're in the lab and actually amplifying this, we're joking that this is like the most pirated book on the planet. Just by doing a simple PCR amplification, you can get millions to billions of copies of this. And so essentially you can amplify and distribute this for free. And this is another kind of example of actually now encoding kind of movies and other things. So this is one of the first movies that's actually recorded in history, and then they've been able to encode that into a GIF. And using the DNA to be able to actually store this information and be able to use sequence to recover that with pretty high fidelity here. And this this other example I want to highlight is something from last year where scientists have been able to combine this idea of DNA synthesis with 3D printing of actual materials. So this is a buddy that has has a polymer, and within the polymer is embedded DNA sequences, right? So these DNA sequences, you can break up the polymer by melting it and extracting the DNA. When you decode the DNA, that gives you the instruction to make a file, a CAD file that goes into a 3D printer to remake the bunny, right? And so this idea is now you can get physical materials that give you all of the information that's necessary to reconstitute and rebuild this physical material. And you can imagine why this could be quite paradigm shifting in terms of how you think about, not only information, but also physical things that contain information. Now, this is obviously in an inert material, unlike a cell in your body, but it has the same type of framework by which you can now start to think about, oh, can I get my iPhone to print another iPhone in case my iPhone breaks?
Hey, Harris,
I see the chat is blowing up, so I definitely want to take a pause here.
Good. Yeah. Do you folks want to ask their own questions out loud, or would you like for me to read them? I know Jacqueline's sound is off, so I'll go ahead and read hers for now. She says, how easy is it to access the information back after you've encoded it? And if the information, or if DNA is information, how can it take in more information? How does it differentiate between the two?
Right, great question. So, to access the DNA is actually quite easy. It's you can specifically amplify subsets within a large pool of DNA, and by essentially this process of molecular hybridization, where you can take a very defined sequence, match it to something else that's stored inside this pool of DNA, and then you can then amplify that. It's almost like there's a molecular way of querying information within this pool of data that's stored in the form of DNA. And there's different ways that you can start to do this, and with new enzymes such as the CRISPR-Cas system, you can potentially even have better specificity of this. But to be able to extract this information is actually relatively kind of trivial comparatively. And the question about adding more information, the nice thing about this is that you can always just add more to the pool, like the existence of the existence of data within this within the pool of DNA is already there, and you can always add more information to that pool, or you could just have separate blocks of this. And so you can imagine having different tubes store different unit, volume 1, volume 2, volume 3 of something. And so you can always mix and match. You can kind of amplify them in different ways. The nice thing about DNA is essentially is that it's quite kind of, it's essentially just droplets of liquid. And so you can kind of, manipulate them as you will.
And Will, are you in a position where you want to unmute and ask some of your questions?
Hello.
Yes.
I don't have any questions.
Oh, I saw that you asked, did the GIF evolve?
Oh, yes.
Yeah.
Do you know if you can, like, is there a phylogeny tree for this?
So maybe you're asking whether these are static images or they're continuous images. Is that
No, I mean, if each frame evolved, then wouldn't they have a phylogeny?
Yeah. So here the information is static in the sense that most of the time DNA doesn't change unless you're copying. The most frequently DNA is changing because you're making a copy and then you're making some mistake there, right? I would say that's also true for like hard drives, right? And it's just that there's kind of error checks, checksums along the way that helps you with that. And people have developed kind of error checking algorithms to kind of reduce the error rates of these things. It's an imperfect science still, and the major barrier right now is still cost. The cost of synthesizing DNA at the scale that can compete with current storage, information medium is we're still, I would say, 4 or 5 orders of magnitude off. But I would say that this is also something that people have only started to really explore over the last five or 10 years compared to this is the early days of data storage in the silicon kind of era. Anything else we should cover before I move on here?
Yeah, I think Lucas has a question. Lucas, do you want to unmute?
Yes, so I was just wondering, I don't have any wet lab experience, but when I'm thinking about it, I have a USB drive, I plug it into my computer and I can transfer data like, gigabytes within seconds. And I feel like in a wet lab, working with DNA, this is more like on the scale of hours. So how fast can this technology like get so that it's like really, practical in everyday life?
Right. So I think probably the most relevant market for this is archival storage at the current moment. It's not going to be competing with real-time access. It's still much slower, right? To be able to print the DNA, to be able to amplify it, to be able to read it. But if you have information that you want to do cold storage, that you want to let's say it was your tax records, right? Or something where you're going to access not very frequently, but you would love to have a copy of something, that's what you want. So, for example, the Motion Picture, Association of America was approaching some of these DNA synthesis companies and storage companies to ask, well, like, could we go and archive our entire library of movies from, back from the early days, because now they're all in kind of film that's about to degrade physically, right? And so you want to be able to store that. And so that's what, I think the most compelling application for the next period is going to be, until we figure out a way that we can really, get this very quickly. But it's mostly, I think, going to be archival storage applications.
To kind of play around in that space, I'm under the impression that like nanopore sequencing is going to be the way to read this type of DNA, and it reads at 450 bases per second is what I'm getting. So that'd be like saving at or reading your file at 450 bits. But since DNA is like 4 bases instead of 0 or 1s, it's probably significantly faster. But maybe you're talking about a kilobyte per second right now.
Yeah, and that is a USB pluggable sequencer. So yeah, it's got some really interesting parallels there. Let me why don't I move on, because I think we still have a few other things to cover here. But yeah, if you have other questions, we'll circle back toward the end here. Okay. So the next thing I wanted to cover is this idea of trying to reprogram or edit the genome, in this case through total synthesis, to get life with completely new properties. And so we've been working on this in the lab, and also in George Church's group when I was a graduate student for a long time. And this idea about essentially getting cells to be able to kind of have alternative ways to encode its information, such that you can now incorporate new chemistries in them, that you can then have them be resistant to, for example, viruses that is trying to infect it, and for it to be contained in a way that may prevent its kind of biological escape into the environment. And so, for example, you can potentially add new letters to the protein alphabet. What I mean here is that this is the canonical way that you turn a piece of DNA into actual proteins, right? This is how cells essentially map the information that's stored in a piece of DNA into proteins through these codons, and you have 64 different codons encoding for 20 amino acids. But if you have the potential possibility of adding more amino acids by just shifting around these assignments, And so you might imagine, well, what would you do with these new kind of letters in the protein, alphabet? For example, you can add in these new amino acids that allow the protein to be really good binder. So for example, in muscles, they have this additional amino acid that's a derivative of something that we normally have that actually allows it to stick to silica and glass at potentially 10 times the amount of adhesion. And this is really great for building new protein-based binding agents. Another example is this modified alanine, which gives it a property of fluorescence. So you can imagine building proteins and that have new fluorescent properties that you can use either for imaging, for different assays, and then potentially building for different kind of applications. And then finally, here's an example of something where you can do pegylation, right? So this idea, what pegylation is essentially allowing a protein to add additional pegylated molecules to it, such that it becomes more resistant to degradation by other enzymes. And why would this be interesting? For instance, if you take a therapeutic a molecule, and if you can pegylate it, now you can have a much higher half-life within the body, so it can circulate within the bloodstream a lot longer, and therefore you can now have a sustained release and maintenance of this drug within the body. And so those are kind of some examples of how you can, by changing the basic kind of properties of proteins, be able to build kind of completely new kind of applications. And that's enabled by the fact that you have genomes that are essentially recoded to allow incorporation of these new alphabets. Another example is this multi-virus resistance. And so in the space of manufacturing, people are exploring the use of microbes to produce lots of very valuable chemicals, from bioplastics to biofuels to fragrances. And this is one example, which is 1,3-propanediol, which is an additive that's actually normally extracted from fossil fuel. And this gets back to this question of green chemistry. And it's used in a lot in biocomposites, in adhesives, in tape, in coating, in paint, and it's been essentially made now in a microbe, in a bacteria, and so they built essentially very large scale, hundreds of $1 million scale refineries to essentially fermentation to essentially produce this using a particular bacteria. One of the big challenges is that when you have bacteria that's making this stuff, they could get contaminated with viruses, and so that's actually a big problem. That if you have a contamination of a virus that's attacking this bacteria, now you have a loss of production of these chemicals, and so building cells that have multi-virus resistance may be really important from a bioindustrial perspective. And this is a product that has hundreds of millions of dollars in sales every year. And so this is something that is both important as well as something that we could work towards. Let me talk about the pandemic, because I think that's something that is very something that's on our minds, have been on our minds for a long time, for the past year or so. DNA synthesis certainly has a role in this, and both in terms of research and in terms of how we kind of think about the potential, biosecurity and the risks associated with technologies in DNA synthesis in kind of emergence potentially of pandemics. So this is a paper that was published recently describing the synthesis, the total synthesis of the SARS-CoV-2 genome from scratch. And they outlined essentially the timeline of how they did this, mapped to the timeline of when kind of the early descriptions of the trajectories of the pandemic. So some of the first cases was described in early December, and it was kind of identified by the end of the month as a virus a new virus, and it was confirmed by the CDC. And by January, about 5 weeks later, 6 weeks later, they had the whole genome sequence. And over the next few days, more genomes were released. And this group decided to actually generate a version of this in their laboratory, and for a very relevant and important kind of study of the process of this virus. But essentially, from the very get-go of having the sequence, they said, okay, let's see if we can synthesize this. And using DNA synthesis over the course of essentially a month or so and change, they were able to get the full length sequence completely synthesized and reconstituted within a cell by February. And they started this basically within 1 month, they were able to do this. And this kind of speaks to the capacity of this technology now, that you can essentially get that sequence from somewhere and reconstitute this virus elsewhere. Now, why were they doing this, and how were they doing this? Right. So this, if you are not familiar, the SARS-CoV-2 genome is about 30 kb, so 30,000 bases, which is not too small, but not too big. And they certainly were able to do this in the matter of a month. And this is just kind of the different components of the genome and mapped to the function of the actual virus here. And what they were able to do is to first synthesize different chunks of these, and they made 9 different chunks. And then they put that into the yeast, and then they had the yeast assemble them into a contiguous fragment that then they extracted out of the yeast, cut it in one side to linearize it. Because this virus is an RNA virus, and what they made was in DNA, they need to turn the DNA into an RNA, which they did in vitro in the laboratory, and then they infected cells to generate live viruses. What they were trying to do was actually not to make just the virus, but they wanted to make a virus that contained a GFP in it. So they added this GFP, the green fluorescent molecule. And why this was interesting was that now you can use this as a potential biomarker to be able to study both the viral cycle of this virus, but also use this potentially in development of different antibody treatments and ways to kind of study it in vitro in the laboratory. But as you can imagine, this is quite alarming, that the fact that you can start to be able to do this. And these were scientists that had rigorous experimental protocols that had to be approved by the institutions themselves to work in a very kind of defined biosafety environment to be able to kind of do this research. But technology is out there. Another example of using this type of technology is to actually get down to very specific regions within, for example, the virus. So we know, for example, this RDP domain, which is a domain on the spike protein of the virus that interacts with the human receptor during the initial steps of infection. The variation within this domain oftentimes is the big determinant of its specificity in terms of whether it for example, attaches or infects humans or other mammals. And so people have sequenced lots of different domains of these, and people were trying to essentially ask the question, do these domains from other, for example, other viruses, of other coronaviruses, could those also interact with the human receptor, and to what level of specificity does it do that? And so people essentially have the capacity to be able to test this now by synthesizing kind of fragments of this in DNA form, put that into a viral genome, and be able to infect this in a cell in a dish, and be able to study that. And of course, this is going back to this question of gain of function research, safety oversight of this. This is not the first time a viral genome has been synthesized from scratch. About 5-6 years ago, there was an example of a group that resynthesized or synthesized a avian flu virus that was very similar to the 1918 flu. And the argument here is, well, maybe we need to be able to do kind of these gain of function studies to understand kind of how these viruses have evolved to infect humans, and what are the risks associated. We'll be able to predict the next kind of pandemic. One of the things that I would say, I would comment about this is that, the fact that we have been able to kind of make a vaccine so quickly was the result of the fact that people have been studying these viruses since the 2003 SARS first iteration of this virus, which came from China, and so which at least stemmed from had the major outbreak there, and so that speaks to the fact that in order for us to have rapid response, we have to have fundamental research knowledge, and that requires potentially years of study to be able to build up proficiency enough to be able to fight a pandemic very, very quickly. And so it's not something where immediately we're going to know this information. The fact that, for example, the vaccine especially the mRNA vaccines that were developed, they had specific designs in how they configure the spike protein that was based off of a very recent set of scientific results, and that made the efficacy of the virus much better. Speaks to the fact that this iteration loop is really important. And so I'm going to take a pause here to have people absorb this, and then respond to any questions people have, because I'm sure people do.
Do you want to throw out the poll right now, Harris?
Yeah, why don't we throw out the poll while I take some questions?
There's a link to the poll in the chat, or to a question in the chat. Should we be doing this kind of research and gain of function research for viruses? Oof.
Was there any specific questions here? There's a question, Gwen had a question of why resynthesize it and not just add something with the CRISPR. So that's a really interesting and important point here. So especially early on in something like the pandemic, the physical material itself is not accessible to people, right? So we only had, the DNA sequence from someone sequencing it, in a database. So you don't have the actual virus, in hand. If you wanted to study it, you wanted to develop an assay, et cetera, et cetera. So the ability to reconstitute that in the laboratory at any given location is important. And so, that's essentially why, having the capacity to synthesize it is really compelling. But also, of course, there is a ton of risk associated with that.
Isaiah asked a question too about viruses and genes. Isaiah, do you want to ask your question, or would you like me to ask it? How are you feeling?
Oh, well, Will seemed to answer it in the chat.
Oh, cool. Okay, great.
Thank you.
Yeah.
DNA versus RNA, what's the difference? Right. Yeah. So DNA is generally the kind of the permanent storage medium for cellular life. RNA is much less stable than DNA, although some viruses are completely RNA-based viruses. For example, the SARS-CoV, the flu, so those viruses actually, in order for them to replicate, make copies of back into DNA so that then they can make more RNA. And so, but in general, RNA tend to be single-stranded, so they're only one strand versus DNA, which tend to be double-stranded. So that's the kind of the general difference between them.
I have a question.
Yes, please.
Yes, what species of microbes do you deal with?
In our laboratory, or just in general by the field?
In general.
Yeah, so people have been working with all kinds of different microbes in the environment, whether it's soil, whether it's human associated. A lot of these organisms is also not very easy to genetically manipulate. So I think there is probably, people think that, oh, you have a microbe, you could go and do that. Actually, it's actually very hard. Most of the microbes people have a lot of experience with tend to be things like E. coli or yeast, which are not going to be so harmful than a pathogenic bacteria. And so, yeah.
I have a question, sort of a biosecurity related open science question. Is there any sort of discussion happening about biosecurity controls on genomes? If you can just synthesize something from the genome, is there any sort of control about who has access to the genome of say the 1918 avian flu virus? I know that SARS-CoV2 genome was open access, put online, and that was hailed as a really great thing. But is there any discussion about controlling the genome as well as actual samples of viruses?
Right. Yeah, I think it's going to be, it's generally, tough to, if you can sequence it, people will sequence it, right? I think this is the thing, right? And so sequencing at this point is cheap enough and universal enough that people could do it very quickly. And so whether that information gets shared online is a matter of question, right? And there's certainly thoughts about whether you should be sharing some of these sequences online. But once the genie is out of the box, you can't put it back in. And so, the sequence for the 1918, flu is out there, right? And so the question is whether we, how open should we be about the science? And, it's a hard question because the more eyeballs are looking at it, the more we can advance this. And actually, that was the way that we were able to kind of address the pandemic was through having many scientists working on the same problem in different ways. So it's going to be hard to have kind of information restrictions in that way. So. But certainly there are probably certain, pathogens out there that, we are holding in restricted places that where that genome sequence is not available, right? So, maybe the anthrax, strain that's housed in a level 4 biosafety facility, they're not going to necessarily put that information out there, and they probably shouldn't, right? Because right now there is no pandemic, and so that information shouldn't be out there. But if there was a pandemic, maybe people would need to know this information to be able to address that, right?
I also
This leads me to another question. I noticed that there have been many ancient civilizations and those people kept dying out. So I was wondering, can we evolve faster than we can create? Sorry.
Yeah, that's a really interesting question. Maybe I'm gonna hold off on answering that until the end here. But I think we are definitely at a point here where DNA manipulation technologies could be outpacing some of the emergent. The whole point here is that if we can stay ahead of the pandemic through engineering kind of synthesis and editing efforts, that we could stay one step ahead of these pandemics. It's almost 8:30, so I'm gonna keep going here, and still have a couple other topics to cover here. In terms of beyond Earth, there's also applications of DNA synthesis in kind of where we want to explore or expand life to other planets. In terms of terraforming entire ecosystems, for example, on Mars, and beyond. And so there's a recent book by one of my colleagues at Cornell, Chris Mason, about the next 500 years. It's a really interesting read if you're interested, about kind of his perspectives on what are the technologies that are necessary for us to kind of bring life to new worlds. And in the book, it actually describes one of the projects that we're working on right now in the laboratory, which is kind of the segue here, which is, if you think about life, right, it requires essential elemental molecules like carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. And these go on to make things like carbohydrates, like sugar, nucleic acids, like DNA, RNA, amino acids, that make up protein, and lipids, fat. And these together make up kind of life on Earth. And if you actually look at metabolism, this is a map of all metabolism that we know of. And mapped on here in red are all the metabolic processes that humans are able to perform based off of its genome. And you'll notice that some of these are actually missing, like specific edges here. So some of these nodes, which correspond to specific molecules, don't connect to each other. And it's because we have incomplete or missing pathways to be able to make some of these essential building blocks. So as a result, we essentially get that from nutrients that we eat, whether we're on Earth or if you go into space, you're going to have to bring that with you, right? One of these examples is an essential amino acids. So for example, you need to eat 9 essential amino acids as part of your healthy, balanced diet, right? And this is the World Health Organization kind of guideline for how much you should be getting out of your food. But where do these come from? They come from essentially trees and other microbes. And that's actually quite amazing if you think about it, that this essential metabolite that's required to make you that doesn't get made by your body. It gets made in a tree somewhere. And through the food web, it eventually makes it into you, right? If you're thinking about trying to go to another planet, maybe you can't afford to bring a tree or bring the ecosystem of all these things. And so the question is, what would you pack on your trip to Mars or Alpha Centauri that will allow you to be able to be minimally sufficient but completely sufficient in your journey there and to be able to sustain there in the future. So it turns out that the ability to make, for example, an amino acid in these pathways existed in a variety of different organisms, but actually it's lost in things like metazoans. So humans, for example, they can't make these, as well as rhinos and sharks. And so basically all advanced animals actually can't make this, which asks a very kind of interesting question. Why have these things been deleted from our genomes when it's such an essential part of our metabolism? But actually, there's lots of other pathways that we could potentially transplant from other organisms into the mammalian genome. So this is just a very incomplete list of 200 plus genes that you can potentially import that make essential fatty acids and vitamins and so on. So we have this project in collaboration with Jeff Buka at NYU on trying to take organisms that naturally produce all of their metabolites in terms of amino acids and put them into mammals that are not able to do this. And we're not, of course, doing this in a mouse yet, but we're interested in doing this in a cell in a dish to just test whether this is possible. And so the idea is that we would go in and bioinformatically mine the pathways that are missing, be able to design them on a computer. We can do gene synthesis to assemble these from small fragments into bigger fragments, for example, in a yeast, and then put that into a cell line, a mammalian cell line that then we can assay for different, functionality to actually see whether it's actually making these metabolites. And so this is just an outline of all the different pathways that we might consider here. For example, histidine, the aromatics, the branching amino acids, and so on and so forth. And the idea here, of course, is that if we could start to make these essential metabolites, we can now get self-sustaining, mammalian cells that could be able to grow on very elemental building blocks, where you could literally take, sugar and air, and then they will just make everything on its own, right? And why this may be interesting, for example, if you're trying to make cultured meat, right? You're actually trying to grow meat on a dish, and maybe you're trying to make a burger, kind of the next generation burger. You want these things to be able to be sufficient so that you don't need to add in, these additional metabolites, which both increases the cost of the bioprocessing and kind of makes this really not sustainable if you have to source them from, other places that are quite expensive. And there are many other applications of this in terms of cell line engineering. For example, could you make these mammalian cells that are more resistant to viruses? And again, maybe this is really important in the context of drug production, right? So oftentimes when you make biologics, you don't want the cells that are making your drugs to be contaminated with viruses. So making them multi-virus resistance is really important. You can make them potentially resistant to cancer, radiation, lots of other phenotypes that are interesting. And this is just to describe that, the use of CHO cells, which is Chinese hamster ovary cells, which is a mammalian cell line that we use for some of our studies, is one of the dominant producers of these biologics. So our ability to engineer these cell lines is going to be really important. And using gene synthesis is going to be a path along this line. Other things like agriculture is going to be also important. This is an example of engineered crops. This is the golden rice, where they're able to biosynthesize beta carotene, which is a vitamin A precursor, and this is incredibly important, especially in kind of in underdeveloped parts of the world, where many people have these vitamin deficiencies. And this could potentially save millions of people. I think finally here, we are, as Guillaume had mentioned, the community is organizing an effort. It's an international consortium called the Genome Project, right, to try to synthesize genomes, human scale genomes, by trying to decrease the cost of the synthesis by potentially 1,000-fold over the next 10 years. And so this will require fundamentally decrease in cost, as well as increase in throughput and lots of iterations to design, test, learn, build cycles. Finally, I think this is my last slide here. We are also in a new revolution. I think the next revolution, which is the revolution in machine learning and AI. And so there's been tremendous advances in the space. I think there's going to be lots of really relevant applications in biology. So this is an example of a new algorithm that Google and DeepMind had developed to predict crystal structures of three-dimensional structures of proteins. It was thought that this is something that was not going to be achievable for potentially decades, but within Google and DeepMind working on this for 2 or 3 years, they were able to essentially solve this problem, right? So there's which is amazing. I mean, I don't think many people were really shocked that one could do this so quickly. And this is just one such competition where they could actually, try to evaluate the performance of these algorithms, where 100%, 90% is basically as accurate as what we could measure by X-ray crystallography, which is a golden standard. And just over the last year, they've essentially jumped the performance of their algorithm to the point where many of the predictions are indistinguishable from those that could be generated experimentally. And so this is going to usher in an incredibly new era of bio design, protein design, that I think we're only at the beginning of. So, for people who are thinking about, what area to go into, I think this is a really exciting and incredibly high growth and area. So definitely something to look at. And of course, many general artificial intelligence algorithms are being developed, some of which will be eventually going to be giving this presentation, and it will generate an avatar, and then it'll mine all of the PubMed for the most interesting articles, and come up with a really brilliant presentation to give to you. So that'll be a really interesting feature for that. With that, I'm just going to close and really thank the people in our lab, and thank Guillaume for being a partner in crime in this presentation here. It's been really fun to interact with everyone, and definitely happy to take on any additional questions, and we'll give you the results of the poll also. So